in that diagram, in last previous classes, we have already discussed the erythropoiesis, that is the erythroblast to the mature RBC formation. So now we'll just go through the RBC membrane. So RBC is like a concave RBC, that kind of shape maintain. So take out that zoom to that. So band 3 protein is there. You can see my cursor. That is the band 3 protein, and then acts as a anionic exchanger. You can write down it. Band 3 acts as anionic exchanger. It acts as a anionic exchanger 1. Another important protein, and that is the glycophorin. You can see that it is the glycophorin protein. This protein has maintained the polarity of the RBC. So that maintain the polarity of RBC. And another protein that is present in that is the GLUT1 transporter. So you can imagine here, it's a GLUT1. It's GLUT1 transporter. So another few proteins that are present in that cellular microtubular system, that kind of band 4.1, actin, spectrin, and these are present in that cellular membrane. So in cytosolic portions, the protein of the RBC like hemoglobin, enzymes like pentose phosphate pathway enzymes are also there, glycolysis enzymes are also there, and if you think about the antioxidant, that is the glutathione reductase and the glutathione peroxidase, and that neutralize the ROS production. So next slide, we talk about in cytosolic portion. In cytosolic portion, there is yes. So there is three type of protein. Already I have mentioned the glutathione reductase and the glutathione peroxidase. So ROS is the reactive oxygen species, and this is kind of byproduct of this metabolism. And what happens if the ROS is too much, and that can react the body. It controls the different kind of mechanism like aging. The ROS is important also along with the muscle fattiness. So reactive oxygen species has different role, like you can say the O2 minus or NO minus, that is the nitrous species also. These kind of molecule are super reactive, and to neutralize to that we need some of the enzymes, like you can call the antioxidant, that is the, we have already mentioned, glutathione reductase and glutathione peroxidase. So these are the neutralize that, these two enzymes neutralize the ROS. Okay, so we already discussed the RBC formation and how the RBC forms, and now we already discussed the cytosolic portions in that in that membrane portions of the RBC, and then we go for the protein portion. And now we are discuss the major component whatever present in that RBC. One is the hemoglobin, and another, that's the hemoglobin. And so hemoglobin, it has two parts. One is heme, another part is the globin. So in heme portion, it's like porphyrin ring and Fe2 plus. It's you can call it, it's a prosthetic group. So it's a like porphyrin ring and the Fe2 plus. So this is the part of the hemoglobin, heme, and globin has 4 subunits, and one is the alpha, beta, you call it beta, alpha. So it's kind of mesh network, it's kind of 4 subunit. You can also write down like depends on their subunit, the different types of RBCs are there. So it's generally, it's a 2 alpha, 2 alpha and 2 beta subunit. Okay, so their position can be changed. So in embryonic stage, you can just go through the embryonic stage. In embryonic stage, like from 1 to 3 months, their alpha chain and the epsilon chains are there. 2 epsilon chains are there. So in embryonic stage it will be there, and in fetus stage, like alpha 2 and the gamma 2 chains are there. So this is called the HbE, and it is called the HbA, fetal hemoglobin. In from birth stage, it will be the alpha 2 and beta 2. Another form is there, that is the 97%, that is called the hemoglobin A, HbA. HbA, it's within the 97%. Another chain is there, that is called the alpha 2 delta 2, that is the less than of 3%. So they are the types of the RBC, and the chain depends on the physiological condition, and from the embryonic stage the different physiological conditions are there. So that's why their chain are also different. In that particular time, their delta epsilon subunit of the hemoglobin and alpha subunits are present, and during the fetal stage it will be like the alpha 2 and the gamma 2, that is also called the HbF, and from the birth stage that will be the alpha 2 and beta 2, and HbA is the 97%. The more, that is called the hemoglobin A, hemoglobin is more after birth. Now is the gamma stage, that is the fetal stage. So this expression is controlled by the locus control region. So has this the gamma 2 in fetal stage is more affinity over oxygen. So that is the more affinity to O2. So now we can go through, okay, these are the section. Now we'll go the how many one hemoglobin, how many oxygen can bind. So like you can write down. So this is one block, and we'll take 4. So you can just take O2, O2, O2 and O2. So first you will write down Hb plus O2, that form the oxygenated Hb, hemoglobin. That is the Hb4 plus O8. That is the main position of that. Now if hemoglobin bind with CO2, which is known as carbaminohemoglobin, and if Hb has higher affinity with carbon monoxide, that is known as carboxyhemoglobin. And the most stable form, if it is formed with that CO, that is the more stable. That's why it acts as a poisonous, and most of the time it is irreversible. So it's a irreversible. So it's kind of statement you can already remind it. Now affinity of the oxygen can changes during the pH changes. So now let's written down the different affinity. Affinity of O2. So if hydrogen ion concentration is increased, then HbO2 affinity will be decreased. Hydrogen ion concentration increase, it means pH is decreased. If pH is decreased, that means environment is more acidic. So in acidic environment, hydrogen oxygen affinity is decreased. That means if we take that block O2, 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 affinity decrease, that means O2 get released or like not bind to that region. So if acidity is increased, let's talk about the another scenario, like H plus ion is decreased, that means pH is increased, that means this is the basic in nature. So in basic condition, what happened there, that is called the in that called the alkaline pH. In alkaline pH, affinity is increased. If affinity is increased, then oxygen will attach with the hemoglobin. Let's talk about the other sections, that kind like the nitrous and other oxidizing compound, and if it is that the Fe2 plus, if it is the oxidation is there, their electron will be released and it will be like Fe3 plus, that is the ferrous to ferric. And if Fe3 plus is there, and then phosphorine, if it is bind phosphorine and globin, and it will form the mate hemoglobin. So this mate hemoglobin cannot transport O2, cannot. So mate hemoglobin cannot transport the O2, and if it is happen, that is the unloading of the oxygen or unloading getting stopped. So affinity increase with the O2, and that means body turns the blue color. If mate hemoglobin, then body turns blue color, blue in color. So these medical conditions or these conditions also known as cyanosis. Hope that everyone will understand that if the Fe2 gets changed and oxidation is more occur, then the Fe3 plus form. Now we'll go the maturation of the globin chain. So can also affect the affinity of the hemoglobin and the oxygen. So like globin chain maturation also involved in that process. The simple substitution, most common type of the mutation is there. If the purine to purine modification or substitution, or the pyrimidine to pyrimidine substitution, like some of the transversions are also there, like purine to pyrimidine transversions, like the adenine guanine to the thymine and cytosine transversions is there. Then the changes of this particular protein is there, like the mutation in alpha chain and the mutation in the beta chain. Let's talk about few words. Mutation in alpha chain. What happened? If mutation in alpha chain, it is called the alpha thalassemia. And if the mutation in the beta chain, that will call the beta thalassemia. So what happened? We know in the 6th codon, like the GAG. In GAG, that responsible for the GLU, and this is the glutamic acid, that is a negative charged amino acid. If the mutation happen in that specific region, if in that mutation happen, what happened? It's from the GUZ. Write down it. It's getting changed. What happened? The glutamic GLU turns into the VAL. VAL means valine. But another nucleotide, and it is the neutral nucleotide. It's a neutral. So if amino acid get changed, that means their property will change. The RBC shape changes. So what happened? RBC shape changed, and it formed like a sickle. So that is called the sickle cell anemia. So in continuous lecture, I have already discussed the pernicious anemia, also discussed the mate hemoglobin type of anemia, and discussed the sickle cell anemia. So these 3 type of anemia I already have discussed. So let's talk about the another mutation. Okay, let's go through. Then in glutamic acid to valine, valine mutation, that is the HBS, sickle cell anemia. If glutamic acid GLU to lysine, then it is called the HBC. That is called the HBC. So that is that all are discussed in the 6th codon region. If we will talk about the 26th codon region, So what happened in 26th region? Lysine get changed to the glutamine and then it's from the HBE. So all about changes of the mutation that can changes the different type of the protein. Now